Hello learners, so today we're going to take up chapter 9 biomolecules, which is a part of biochemistry. So let's get into the chapter. So the topics to be covered in this chapter are introduction, how to analyze chemical composition, primary and secondary metabolites, biomacromolecules, proteins, polysaccharides, nucleic acid, structure of protein and nature of bond linking monomers in a polymer. So let's get by the introduction. Biomolecules is a chemical found in all living organism, or all the carbon compounds that we get from living tissue can be called as biomolecules, right? Now I have a question for you all. Are all living organisms made up of the same chemicals? Yes. Suppose we take an example of earth crust or living tissue. Any living tissue, right? So we being human, we take elements from plant take these elements from soil, so we both taking these elements from the same source earth, so we have like the same elements. So they both are made up of the same element but different in amount and compound. However, carbon and hydrogen is higher in any living organism than in earth crust. Also, there is a table given in your NCERT of composition of earth crust and human body, which you don't need to remember, but you just need to know the higher percentage series of elements in human body, which is oxygen 65, and then carbon, which is 18.5, nitrogen, which is 3.3, calcium, calcium, which is 1.5. Now we'll perform a chemical analysis of organic compounds, where we'll take a living tissue, maybe a leaf or a piece of liver. We'll grind it in mortar and pestle by the help of trichloroacetic acid. We'll obtain a thick slurry. Now we'll strain it out through a cheesecloth or a muslin cloth. Then we'll obtain two fractions. One is called filtrate, or more technically acid soluble pool, and another is retentate, which is like acid insoluble pool. Acid insoluble pool has biomacromolecules, which are big in size, which are stuck on the surface of strainer, and in acid soluble pool we have biomicromolecules, which are inside of the beaker. You can see in the picture. In acid insoluble pool, we have biomacromolecules whose molecular weight is more than 1000 Dalton. We have mainly four types of organic compounds in it, which are protein, polysaccharides and nucleic acid. These compounds are polymeric substances. Here we have an exception of lipid, which is non-polymeric substance made up of simple molecules. But the twist is, neither its weight is more than 1000 Dalton, nor it's a polymeric substance. Then why we have lipid in biomacromolecule? Because lipid forms aggregation, which we'll discuss later on. In acid soluble pool, we have biomicromolecules whose molecular weight is less than 1000 Dalton. Other than that, we have thousands of organic compounds found in acid soluble pool only. But you don't need to remember the names of all of them. But for your information, I'll mention 7 to 8 names, which are as follow: ions, amino acids, monosaccharides, nucleotides, nucleobase, nucleosides, disaccharide sugars, and fatty acid glycerol. We have already performed chemical analysis on organic compounds, but now in this activity we'll see how inorganic analysis take place in living tissues. For that we will need a piece of liver or a leaf, any of these two. This is called fresh or wet weight when we have moisture in it. Then all the water will evaporate by the process of evaporation, then we'll get dry weight. Then we'll burn this dry weight leaf so that all the carbon compounds are oxidized to gaseous form. CO2 and water vapors are removed out, and then remaining will be ash weight. The ash contain lots of inorganic elements like calcium, magnesium, sulfur, phosphate, water, and many of these. If you remember, then these ashes contain some inorganic elements are also seen in the acid soluble pool, right? You can skip this part if you already know. These are the organic and inorganic chemical compounds from the chemistry point of view, which is a protein consists of 10 to 15%, nucleic acid 5 to 7%, carbohydrate 3%, lipid consists of 2%. It varies accordingly, right? In organic part we have water, which consists of 90% almostly, and salt is 1%, ions And in our cell protoplasms, these salts are present in the form of ions, which is sodium and chlorine. As it is clear now, the decreasing order of organic compounds in protoplasm, starting with protein, then nucleic acid, carbohydrate, and then we have lipids. I hope you can easily identify this functional group from the previous session, but let me recall it for you. We have amide group and amine group, which are nitrogen containing groups, and we have thiol group also, which is sulfur containing group, and other than that all other have oxygen content in it. So let's begin with amino acids. Amino acids are the organic compound containing an amino group which is basic in nature and an carboxylic group which is acidic in nature. Are they substituted on the same carbon, which is alpha carbon? Hence, they are called alpha amino acids. They are substituted methanes. Why? Because when you take a structure of methane, we have four different functional groups. As I already said, there are four substituted groups occupying the four valency positions, which are hydrogen, carboxylic group, amino group, and one variable group, which is R group. Okay, so on the basis of the nature of this R group, there are many amino acids. Approximately, there are 300 amino acids are known to be exist, but out of which only 20 types of amino acid which help in the protein synthesis. That is why they are called proteinaceous amino acids. Okay, so we have 10 essential amino acids and 10 non-essential amino acids. Essential amino acids are those which are taken from the diet, which we can't prepare in our body by our own self. So the name starts with arginine, isoleucine, valine, lysine, threonine, histidine, methionine, phenylalanine, tryptophan, and leucine. Hence, these dietary proteins are the source of essential amino acids. Out of these 10 essential amino acids, 2 amino acid arginine and histidine are synthesized in animal body, but in very less quantity. But these amino acids are required in very high quantity. Okay, so these two amino acids are also known as essential amino acids. Other than that, we have non-essential amino acids made from the essential amino acids directly inside our body. So the name are arginine, aspartic acid, glycine, alanine, serine, cysteine, glutamic acid, asparagine, glutamine, tyrosine, and proline. You can see their polarity written on the right side, either it's polar or non-polar. Few of them are glycine, cysteine, glutamine, tyrosine, and proline are semi-essential, needed in less only. But you don't need to remember these names, right? But yeah, you must need to know all these 20 amino acids name at day or night. You need to remember it at anyhow. Classification of amino acid on the basis of R group and different pH. So in acidic pH, we have 2 carboxylic group and 1 amino group. You can see in the example of glutamic acid and aspartic acid. This is star point. Only acidic amino acid can form amide. See the example. Like if we react NH2 with the extra COOH group of glutamic acid or do the same with aspartic acid, then it will become asparagine and glutamine. In the neutral amino acid, it consists of one amino and one carboxylic group because of this deuter ion or amphoteric nature of the molecules, which shows the both acid and basic properties. Molecules have equal positive and negative charge, which creates a special point called isoelectric point when a specific value of pH at which amino acids neither move towards cathode or nor move towards anode, which is isoelectric field applied in it. Even you have done questions related to pi or to find pi in your chemistry sessions. And we have glycine as an exception because of no chiral carbon in it. I guess we already know that what is chiral carbon. When a carbon atom whose all 4 valencies are satisfied by different atoms or molecules. But here in the structure of glycine, you can see we have 2 hydrogen bonds. One we have already and the another one is in the R group. So because of that, it is optically inactive and the only simplest amino acid out of the 20. Unlike acidic amino acid, we have some other basic amino acids: neutral, sulfur containing, amino acids, alcohol and aromatic. We have a few examples given each of these and we'll discuss. Let me take you through the examples, then we'll get a clear observation of the examples for better understanding. So in the basic or alkaline pH, we have lysine, histidine or arginine, where we have two amino groups and one carboxylic group. In for example, lysine, we have two amino group, and in histidine we have a ring structure. You can see lysine and arginine are in histones. DNA is basically packed within histone protein. We'll read all these things in cell chapter. Other than acidic and basic amino acids, rest 15 amino acids are considered as neutral amino acids, with examples like sulfur. Sulfur containing amino acids are those which help to make disulfide bridge, for the stability of the protein and covalent bonds. Examples are cysteine, cysteine and methionine. Amino group is part of amino acid where it's a part of secondary amide rather than primary amide, where one of the hydrogen atom is replaced by a side chain forming a ring structure. And proline is the only example of amino acid. Cysteine is the only SS-thiol group containing group amino acid, and cysteine has disulfide bond in it. Just because of this heterocyclic ring in the structure of proline, it's also called as heterocyclic amino acid. Moving towards alcohol, OH group, we have serine with hydroxyl R group, threosine which is again cyclic ring structure, and threonine with the hydroxyl R group structure. And aromatic group contain aromatic rings in their side chains. Examples are tyrosine, phenylalanine and tryptophan. There's a fact about tryptophan is the most complex indole ring structure, and even is the most complex amino acid also. It's a precursor of auxin and helps in the synthesis of plant growth hormone. The information about the amino acid content is also going to help you in the upcoming nutrition lessons. Let's begin with protein, which are biomacromolecules and hetero-polymers, which means they are like each protein is a polymer of 20 different amino acids, like it depends, which you already learned in the previous slide. Now moving towards, from the biological point of view, protein performs many functions in the body. Now we'll see what are peptide bonds in amino acid. So basically, peptide bond is a type of covalent chemical bond that links two amino acids together in a protein or a peptide chain. As you can see in the image, the carboxylic group of first amino acid reacts with the amino group of second amino acid, any another amino acid, and a molecule of water will release out through dehydration reaction, where water is produced as byproduct and dipeptide chain will form. Dipeptide chain is basically simplest peptide chain. It consists of two amino acids linked by one peptide bond. Always remember this thing. When more than two amino acids link together by peptide bond, they'll form polypeptide chains, which is a long continuous chain of peptide bond. In amino acids, variation depends on its side group, but in proteins, variation depends on the sequence of amino acid or the configuration of protein molecules. Let's start with the classification of protein on the basis of its structure. So there is a fact. Before starting, I will like to explain. It will take a few minutes of yours. See, in organic chemistry, the structure individually refers to a molecular formula. You can see, like you can maximum you can say is NaCl, Mg+2 and etc. But in if we talk about organic chemistry, we always write a two-dimensional view of molecules while representing their structures, which is you can take the examples are most famous benzene and naphthalene. Max to max you can draw these like 2-degree structures, but in physics there is a three-dimensional view of molecular structure, while biologists describe the protein structure at 4 different levels, which means 4D structures. You can clearly see the beauty of biology, while no offense although. So the primary structure is a very simple and basic 1-degree structure. As you can see in the picture, the first one is primary structure. Wait, let me draw it for you so that for your better observation and clarity. Primary structure is a linear sequence of amino acid where left end represents the first amino acid starts with positive charge and terminal, and the right end represents the last amino acid which ends at the negative charge C terminal. So when I say 1 degree structure, what comes first in your mind? Okay, take a minute and think on it. See, it is not likely to be rigid rod-like structure, but it is a thread folded in the form of helix. Something similar reminds me of revolving staircase. Yes, that it is. And yeah, only some proteins of the portions of the protein thread are arranged in the form of helix, and also only right-handed helix are observed in the protein thread structure. And also it is the most unstable structure of the protein. It has no importance of its own. You know why? Because it doesn't perform any function. If you may remember, then I said polypeptide may be folded in structure but must be functional. Then it would be protein. Then it would be called as protein. Then what functionality here means is to perform some biological functions in our body. If you already know that protein is the main building block elements in our body, then how if someone is not even functional will make the building blocks in our body? Doesn't it sound so weird? That is why these 1 degree structural proteins doesn't perform any well specific functions in our body, but the only function is to determine the positional information to the sequence of amino acids to tRNA, which means which is the first protein, which is the second protein, and so on. And the best example would be newly formed proteins on ribosome have these primary structures only, if you ever noticed. Moving towards the secondary structure, which is 2 degree spirally coiled. These structures are basically of the two types, as you can see in the picture, alpha helix and beta plated sheath or helix again. So alpha helix structure are with the right-handed rotation and peptide bonds are mostly in it. And the intermolecular hydrogen bonds between these two amino acids of the same chain. You can see the repetitive structure of hydrogen bonds in blue color structure. Examples are keratin and myosin. And another one is beta plated sheath, which is similar to zigzag structure and peptide bond in it again, and it has intermolecular hydrogen bonds between two or more than two peptide molecules. And the example is fibrin silk because of its fibrous appearance. If you're still confused with the hydrogen bond, that's okay, let me explain it again. Amino acids are linked by hydrogen bond between oxygens of one amide group and hydrogen of another amide group. Don't be confused. See, if oxygen whose electronegativity is more and hydrogen whose electronegativity is less, then there is a weak force of attraction develops between these amino acids. That is called hydrogen bond. Actually, hydrogen bond is not a specific kind of bond, but it is only a weak attraction force. And as I already said, there are two types of hydrogen bond we see. First is intermolecular hydrogen bond between two or more than two molecules in beta plated sheaths, and another is intramolecular hydrogen bond within only a single molecule formed by the hydrogen shifting process. We'll talk more on it on further classes, but till then you remember that RNA is a single helically coiled structure and DNA is a double helically coiled structure. Okay, I'll ask you. Third one is 3 degree tertiary structure, which is more like hollow woolen ball like structure. And as I said, like 2 degree structures are not soluble in water, but these 3 degree structures are definitely soluble in water, like in colloidal solutions. And majorly proteins and enzymes in the protoplasm has these tertiary structures exhibited in it. And other than that, these tertiary structures are absolutely necessary for performing many biological activities of proteins. Long protein chains is also folded upon itself like a hollow woolen ball, giving rise to these 3D structures. Like 2 degree structures, we have again many bonds in it as well. Peptide bond, which is the strongest bond of protein. Second one is hydrophobic bond between these amino acids, which have hydrophobic side chains. aromatic amino acid which you have seen in it. And third one is a hydrogen bond and van der Waal. Hydrogen bond I'll already explain you. And the fourth one is disulfide bridge, which is the second strongest bond after peptide bond. And there's nothing new about it. I've already explained you disulfide bridge in the sulfur containing amino acids, if you remember. These bonds are formed when the between these SH thiol group amino acids, remember. And fifth one is ionic bond. I guess it is not that unique thing for you. We have already discussed formation of ionic bond between two opposite ends of protein molecule due to electrolytic attraction. And the examples are enzyme, hormones, etc. And don't feel sad for yourself. You don't need to remember all these bond names for your board exams, and it is not even in your NCERT. I just explained you for better clarification and for your higher studies if you're gonna do so. And if not, then you can completely avoid it. That's okay. Last but not the least, quaternary structures, which is 4-degree and the most stable structure of protein which I've discussed till now. These 4-degree structures forms when two or more polypeptide chains of those tertiary structures unite by different type of bonds to give rise these 4-degree structures. And for examples, we have different polypeptide chains, maybe similar one and maybe dissimilar one. But in your NCERT, they have given only dissimilar types of polypeptide chain examples, which is hemoglobin and insulin. Let's talk about hemoglobin. They have like 4 different polypeptide chains in it. 2 is alpha subunit and 2 is beta subunit. Combinedly 4 polypeptide chains. And moving towards insulin, we have 2 polypeptide chains in it. One is alpha subunit and another one is beta subunit. And other than that, talking about the similar one examples, we have lecithin and dehydrogenase, which we don't need to understand for now. Okay, so we have the most important functions of the protein from your NCERT table number 9.5, as you can see on the screen. So the first example is collagen protein, whose function is to intermolecular ground substance which provide strength to the bone, to the skin and to the tissues. We have already discussed it in the tissue chapter, and definitely it is the most abundant protein in the animal world, collagen. The second one is trypsin, which breaks down the protein into the smaller in small intestine. And the third one is insulin, which regulates blood sugar level. As insulin is a hormone, I guess these are the very basic topics from the junior classes. Trypsin, insulins, we already know it. Okay, now the fourth one is antibody, which fight against the infections. Fifth one is receptor. What kind of receptor? Sensory receptors. Act as smell, taste and hormones. The sixth one is GLUT4, where it stands for glucose and T is for transport. It enables the glucose transport into the cell. We'll see this in cell chapter also. And these are the only 6 which we have to understand, but I have few more for you, which is like hemoglobin, transport oxygen into the blood. Amylase break down the starch into the sucrose, which is an enzyme. We have keratin protein, forms hair, nails and the outer layer of the skins, follicles. We have DNA polymerase, helps in the DNA replication, it's an enzyme. And albumin protein maintains osmotic pressure and transport the substances into the blood. And these are all that will be enough for you. Students, there is one more concept of denaturation of proteins, but it is not in your NCERT, so you just not need to know all this. So I'm just skipping it for now. So now we are moving about the another part of this chapter, which is lipid. What is lipid? Lipid is something which is fat and its derivatives combinedly called lipid. Lipids are generally water insoluble, which means they are non-polar. But they are soluble in some organic solvents like acetone, which is in nail paint, and chloroforms, benzene, hot alcohol, ether, etc. Now if we compare lipid with carbohydrates, we'll see that fats and lipids produce more than twice the amount of energy as compared to carbohydrates. And as we already know, animals store maximum amount of food in the form of lipid. In these animals, fats and lipids are present in subcutaneous layer and work as food reservoir, and one more is a shock absorber. So you will see these things in tissue chapter in more detail. And again, second thing is lipid requires also very less space for storage as compared to carbohydrates, because lipid molecules is hydrophobic and really very condensed in nature, so they don't need much space. See, lipids are basically of three types, which is simple lipid, also called as neutral fats. Another is conjugate lipid, which is also called as compound lipids, and last one is derived lipids. So we'll discuss one by one. Let's begin with simple lipids. These simple lipids are the esters of long chain fatty acids and alcohol functional group. In majority of simple lipids, these alcohols are trihydroxy sugar alcohol. Example is glycerol, you will see further. Now what I am trying to make you understand about simple lipids is the ester bond and how they forms. Question is, what are ester bonds? See, when three molecules of fatty acids linked with one molecule of glycerol, ester bonds will form. Such type of lipid are called triglycerides. Okay. Now, three molecules of water released out during the formation of triglycerides. It's a dehydration reaction where a molecule of water released out. So, first is fatty acid. Fatty acids are the carboxylic group attached to an R group, where R group could be methyl, ethyl, or higher number of CH2 group like 1 to 19 carbons. Now we have two different type of fatty acid. The first one is saturated fatty acid and the another one is unsaturated fatty acid. So in saturated fatty acids, they could be with single bond and without double bonds, but in unsaturated fatty acids they are with one or more double bonds in it. As you can see in the image of hydrocarbons, we have only one single bond between the carbons, and in the unsaturated one we have double bonds written over the screen, you can see. Lipids with more amount of saturated fatty acid in it are mostly present in solid forms at the normal room temperature. Examples are like butter, ghee, and in unsaturated fatty acid lipids are present at liquid form at the room temperature. Examples are coconut oil, mustard oil, or any seed oil. So examples are palmitic acid, as you can see written here with structure. It has 16 carbon chains in it, and arachidonic acid with 20 carbon and 4 double bonds in it, and one more thing, including carboxylic carbon group in both of them, as I already said. There are a few more examples of it, but these are the only two written in your NCERT. So let me brief by saying there is like one more classification of unsaturated fatty acid, which is MUFA and PUFA. MUFA basically stands for monounsaturated fatty acid with single bonds in it, which is non-essential fatty acid, and PUFA stands for polyunsaturated fatty acids with the essential fatty acid double bond in it. Another simple lipid is glycerol, whose scientific name is trihydroxypropane, where tri means 3 and hydroxy means OH group, OH functional group. Now many lipids have both fatty acid and glycerol in it. Here the fatty acids are found esterified with glycerol, and by this process we have three different types of glycerols, which is monoglyceride, diglyceride. Alexa, please stop. Monoglyceride, diglyceride, and triglycerides. Very sorry. So in monoglycerides, the structure of one glycerol reacts with one fatty acids by the ester bond. One molecule of water will release out. By this process we'll get monoglycerides, and by following the same, when monoglyceride reacts with two fatty acid, we'll get diglyceride, and the diglyceride reacts with three fatty acids, we'll get triglyceride, which is our common fats and oils. Now these triglycerides are of two types. One is fat and another is oil. So on the basis of melting points, oil have lower melting points usually. Example, you can take anyone like gingerly oil, and hence remains oil as oil in winter. But while talking about fats, they are with high melting points. Examples are ghee, butter, and you can also reverse this whole process. First you need to take a molecule of triglyceride. By adding 3 molecules of water into it, it will break the ester bonds between them. By the process of hydrolysis, it will become into the simplest fatty acids. It will simplify first into triglyceride, then diglyceride, and at the end monoglyceride. We'll also get 3 fatty acids like R1, R2, and R3 functional groups simplified way.
You can see in the yellow or red color we have two different fatty acids with R1 and R2 groups combines with the glycerol, which is in green color. These both are hydrophobic and non-polar in nature. Then we'll add phosphoric acid, which you can see marked in the blue color, with one nitrogenous compound. It could be anything, but the example is lecithin. So the nitrogenous compound in lecithin is choline. So we'll add phosphoric acid with choline. These two are hydrophilic with polar ends. So one specialty about this is a phospholipid is a compound where we have hydrophobic and hydrophilic both parts together in it. So it's also called as amphipathic molecule. Okay. Due to this property, phospholipids form bimolecular layers in cell membrane, which we'll see in the cell chapter. Another example is glycolipid, which forms bond with carbohydrate, and which we'll see in the cell chapter. Now we have derived lipids which are derived from simple and conjugate lipids. These lipids are really complex in structure and nature also. And most importantly, these are not the real lipids because they are not made up of fatty acid and alcohol. Above example is from your NCERT only, but to understand cholesterol in a better way, first we need to know what are steroids. So basically these steroids exhibit tetracyclic structures in it, and on the basis of functional group we have two different types of steroids. Okay, so the first one is esterols with alcohol functional group and suffix ol in it, and the another one is esterones with ketone group suffix on in it. Now moving towards sterols, which are alcoholic steroids, as I said. Now the best example is cholesterol because it's a parent steroid, and it's also called as most decorated micromolecule in biology. And many other several biologically important steroids are derived from these cholesterols. These cholesterols evidently occurs in brain, nervous tissues, adrenal glands, and skin. For skin, I have an example for you, which is in skin we have sebaceous glands in it which secrete some waxy materials, sebum. Now these sebum have 7D hydrocolesterols which occur in skin, sebum specifically, as a provitamin, and on exposure to ultraviolet radiations from the sun it transforms into cholecalciferols, which is vitamin D. Now the next steroids are sterones, which are ketonic steroids. For example, we have sex hormones like testosterone, male sex hormone, and estrogens, female sex hormones, and adrenocorticoids, and some hormones of insects also. Moving towards another important part of this chapter, which is carbohydrate. Simply, they are the hydrates of carbon. What it means? It means any organic compound comprising of carbon, hydrogen, or oxygens with the ratio 2:1 are simply called the hydrates of carbon, with the general formula CnH2ON. But if you see from a practical point of view, the molecules of hydrogen and oxygen can't be equal always in the ratio 2:1, not applicable for derived carbohydrates. So because of this definition considered as an old one, and now we'll see the new definition of carbohydrate, which says they are the optically active polyhydroxy, aldehyde and polyhydroxy ketones, or the compounds which produce such unit on hydrolysis. Okay, poly, which means many, and hydrolysis of OH group. They are instant source of energy in our body with the high content in a balanced diet. In a normal man, almost 65% of energy is available to him in the form of carbohydrates present in his diet only. And those simple carbohydrates with soluble in water and sweet in taste are called sugars. Now moving towards the classification of carbohydrates on the basis of number of saccharine units obtained on the product of hydrolysis. Hydrolysis, which means when we'll break the carbohydrate bonds in the presence of water. We'll see carbohydrates are of three different types: monosaccharides, oligosaccharides, and polysaccharides. Let's begin with monosaccharides. Monosaccharides are the simplest carbohydrate because no further hydrolyzation is needed, and it doesn't even needs any digestions. Only oxidations are required for energy requirements. All monosaccharides are present in D and L form, with the exception dihydroxyacetone, which is optically inactive. And maximum sugars are dextrorotatory, but with the exception fructose, which is levorotatory. Now, all monosaccharides are reducing in nature, or also called reducing sugars, because their functional groups are in free form or in the reactive state. so that we can easily identify or detect these sugars in any mixtures. So on that phenomena, we have a proper Benedict test, which we'll see in the next slide. This property is the basis of Benedict test or Fering's test, used to detect the presence of glucose in urine. See, basically, in normal glucose is absent in urine, but in the diabetic patients it is present, and it can be checked by this test. One thing you should keep in your mind that all monosaccharides show these Benedict test. Why? Because of these free functional aldehyde and ketone groups. All monosaccharides are reducing in nature, so they show this test. Now let's begin with this activity. First we'll take a test tube with some urine sample in it. Then we'll drop few Benedict solution in it. Then in this Benedict test, Benedict solution is used in which CuSO4 is present, where copper is present in copper plus 2 state, and its color is blue initially. Then we will place this test tube on a burner or a tripod filled with water solution inside a beaker, or you can directly put this test tube on the flame directly for 2 to 5 minutes. Be careful while doing this. Now what we'll notice here, the aldehyde or ketone group of these monosaccharides reduce CuSO4 into Cu2O. And in Cu2O, copper is present in copper plus 1 state and its color changes. The results we will get to know the change in color from blue to red depending on the amount of sugar present in it. While initially the blue color had 0 sugar in it, and the end result is is the sugar traces are really high in it with the reddish or brick red color. And after that, coming towards the conclusion, it means we can say that first carbon is reducing center in the case of aldose sugar, while in the second carbon is reducing center in the case of ketose sugar. Sorry. Now I'll see the classifications of monosaccharides on the basis of number of carbon. Well, the generalized formula is CnH2ON, where the formula X is mostly equal to Y, means the number of carbon or oxygen atoms are all mostly same. So now the first example is trioses. Tri means 3. See, number of carbon count in monosaccharides starts at 3 because that is the smallest number needed to form functional group, and they can vary up to 3 to 7. Okay, sometimes 8 or more than rare cases by researchers only. So now trioses is the simplest monosaccharide with the examples glyceraldehyde and dihydroxyacetone. Do you remember the exception which we learned last lesson? It is optically inactive, yes. And then we have tetroses with the example erythroses and eruloses. The examples like glyceraldehyde or erythroses are the examples with ketone group, while the other two are of aldehyde groups. Now the third one is pentose with 5 carbons in it. 5 carbon means furan ring present in linear, chain or furan, 5 membered ring structures also. So examples are ribulose followed by ketose group, if you remember RuBP1, and the 3 are of aldoses group like ribose present in RNA, ATP, FAD, FM and NAD and others. Then deoxyribose, which is an exception, we already know because deoxyribose technically is a reducing sugar, but it doesn't have behave like one in Benedict test because of its structure. If you see the structure here, the general formula doesn't obey its rule. The carbon is 5 carbon, 10 hydrogen and oxygen is only 4. So you can see carbon and oxygen's ratio are not equal here. And other than that, clearly means that deoxyribose lacks an OH group at second carbon. That is the reason why nature selected deoxyribose for the DNA. It's present in DNA structures. And the third one is arabinose, which is present in Arabic gums. Then we have exoses, pyran ring structure with 6 carbon aldoses. Examples are glucose, which is the most abundant organic compound on earth. Other than that, it is abundant in grapes, so called as grape sugar, abundant in blood, so also called as blood sugars, and corn or glucose drip, which is an instant source of energy. Then we have sweetest carbohydrate, which is fructose, more abundantly found in honey and sweet fruits, so called as fruit sugars. And unlike glucose, here fructoses are levorotatory. Exception, then galactose, which is a main component of lactose milk and present in brain sugar, important component of milk, as I said. And galactose is a component of hemicellulose, pectin, glycolipid, which you will see further. Now we have another carbohydrate, which is oligosaccharide, hydrolyzed to give 2 to 10 monosaccharide units or monomers. Now you can see glycosidic linkage bond between these carbohydrates. See, basically monosaccharides are linked together by glycosidic bond formed by the loss of a water molecule between them. Now we'll see there are few different types of oligosaccharides on the basis of hydrolysis. So when we hydrolyze oligosaccharides, we'll get disaccharide or dimers, trisaccharide, tetrasaccharides, but we don't need to understand all these. Only disaccharide is in our syllabus, so we'll discuss only this. Glycosidic linkage always take place between the ring structures of monosaccharides. Here you can see on the screen, I have an example for you of lactose. So you can see monosaccharides like galactose, first one is galactose and another is glucose. So they are linked together by glycosidic bond between them, which is aldehyde or ketone groups of one monosaccharide reacts with the alcohol group of the another monosaccharide, which is glucose. This is how they forms glycosidic bonds between them, and between these bond formation, one molecule of water eliminates out during the bond formation, which is by the dehydration reaction, which I have already told you in the previous sections. And they are water soluble and sweet in taste, so they also called as sugar like other carbohydrates. So the hydrolyzation list begins from disaccharides and goes up to 10, but you only need to understand disaccharides, as I already said. So let's begin with it. See, they are the simplest oligosaccharides because only 2 units of monosaccharides are present on hydrolysis. Okay, so with only 2 units, it becomes too easy to understand. And also disaccharides are the most common oligosaccharides found in the nature. So examples are maltose, sucrose, lactose and trehalose. We'll discuss them one by one. So let's begin. So these are the types of disaccharides, starting with maltose, also called as malt sugar, which is reducing in nature and present in germinating seeds. It is intermediate compound in starch digestion also. And second is lactose, which is called milk sugar, reducing in nature. Lactose is least sweet sugar. Then we have sucrose. In plants, transportation of sugar mainly occurs in the form of sucrose only. Then sucrose is also known as invert sugar. Examples like cane sugar, table sugar or commercial sugar, same. But you will see that cane sugar, table sugar, commercial sugar, these are the pure sucrose, but not the invert sugar, natural invert sugar. So first you need to break down these table sugar or commercial sugar into glucose and fructose units, then it will become invert sugar. And also you need to hydrolyze these cane sugars, then it will become invert sugar. So that is the different thing that will be in the chemistry point of view. So yeah, and sucrose are also the non-reducing sugars. And then lastly we have trehalose, which is present in insect hemolymph, and they are non-reducing in nature. So now begin with polysaccharides. So basically in disaccharides we saw there were only 2 monosaccharide units, but in polysaccharide they are composed of larger number of monosaccharide units, which on hydrolysis give more than 10 monosaccharide units, both monomer and polymers. Okay, now polysaccharides are insoluble in water and do not taste sweet, except the exception inulin. And all polysaccharides are non-reducing in nature. But in a polysaccharide chain, one end is reducing and another end is non-reducing in nature. But that is from the chemistry point of view. Although polysaccharide is just non-reducing in nature. Now, on the basis of uniformity in monomers, we have two different types of polysaccharides to read. First is storage polysaccharide. They are the ideal storage material because they have few properties which they obey very perfectly. So, first one is they are chemically inert and osmotically inactive. Chemically inert means they don't react with anything in the surrounding, which is actually a good thing for a perfect storage material. And osmotically inactive means they don't absorb any water, or they don't absorb or they don't lose any molecule throughout it. And they also present in layers, so they occupy very few space. Now let's move towards our first polysaccharide structure, which is a starch. So these starch are made up of the homoglycan, which means homopolymer of glucose. Now they are the storage polysaccharide of plants and protista. Now you see there are two types of chains, starch amylum chain. The first one is amylose, which is a linear chain, and second one is amylopectin, which is a branched chain. As you can see on the screen, we have their figures, their linear or branched chain figures. Now see, amylose consists of 20% part of starch, while amylopectin is a branch structure, so it consists of 80% part of the starch. So these amylose are unbranched and helix in structure, while amylopectin are unbranched in structure. Now if you can see on the basis of solubility, these amylose are soluble in hot water. That means when these starch form helical structures, so starch can hold iodine molecules in the helical portion, which means they are like blue black in color with a positive iodine test. Now if these amylopectins are we already know these are insoluble and gives red violet color with a negative iodine test. And yeah, of course you don't need to remember these structures, but you can see in amylose we have unbranched structures, and in amylopectin we have branched structures. You can clearly see on the image. And the formation of polysaccharides by the condensation of monosaccharide unit, which means when two number of monosaccharides polymerize by glycosidic linkage, they will lose one molecule of water. And similarly, if there were 3 units of monosaccharide linked together, they will lose 2 molecules of water, and so on. Next is glycogen, which is the another storage polysaccharide in animals. That is the reason why they are called as animal starch. Now these glycogen are the storage form of carbohydrate in animals, and to be more specific, liver and muscles are the storage region of these glycogen in animals. And if you compare in between, liver stores much more glycogen as compared to muscles. They are completely insoluble in water, so does they don't they give a negative iodine test with red violet color. As you can see in the image, there is no helical structure for iodine test. They are branched or highly branched. If you compare with amylopectin, it's really highly branched. And they are also the stored food of fungi. The third one is inulin. They are the storage polysaccharide of plants, mainly flowering plant families. NCERT says they are the linear polymer of fructose, which means they are made up of 20 to 30 fructose units linked together by bonds. Now see, there is a special fact about it, which says they are water soluble polysaccharide used to measure GRF, glomerular filtration rate. See, the roots of Dahlia plant contain inulin in it, which can be used to measure GRF, which is a key indicator of kidney functionality. So basically, what you need to do is, first you have to give the fixed amount of mixture mixed with the water to the patient, and after that a few minutes you can collect the urine sample, which you can directly measure the functionality of kidney by whatever the amount will be there in the urine. Fact behind it is our stomach can't digest these roots of Dahlia and can be easily filtered out in blood. So that is why we use roots of Dahlia for this experiment. Now we have a structural polysaccharide, which is present in the structure or body walls of the organism. Now the first one is cellulose. They're the most abundant carbohydrate or organic compounds with the bonding of homoglycan or homopolymer of glucose. They are the linear polymer of glucose unit, which means they are unbranched or non-helical fibrous thread-like structure. They are also the main component of plant cell wall, which you will see in the cell chapter, and composed of 50% part of the wood and 90% part of the cotton. And it is also used to form rayon fibers, which is artificial silk. This topic is from the next chapter, but I have to include it here just for the understanding. Now you can see the layers of cell wall. Cellulose is in the form of thread, which is called as microfibrils. There are many cellulose chain in a single microfibrils. As you can clearly see in the matrix of cell wall, there is a cross-linking structure of microfibrils of cellulose, which doesn't allow to enter the large molecular harmful bacteria inside the cell. Now we have chitin, also called as fungal cellulose. It is the second most abundant carbohydrate or organic compound found in nature. Now it is homopolymer or linear polymer of NAG, which means N-acetyl glucosamine, where N stands for nitrogen. So it means there is a huge content of nitrogen in chitin. Now chitin is an important component of exoskeleton of arthropods or cell wall of fungi, which means if you add calcium carbonate CaCO3 to it, it will become the exact structure of arthropods or fungi. Do you remember if you ever tried to hit an insect by your slippers, there is a chut sound came out from it. That sound is because of the breaking the structure of the chitin in it. Now we are done with all the biologically important homopolysaccharides. Now we have few hetero-polysaccharides or hetero-polymers. They are of amino sugars or chemically modified sugars like glucosamine or N-acetyl galactosamines, GAGs. Because of that, they are called mucopolysaccharide, which attracts water because they are hydrophilic in nature, and that is the reason why they form viscous material. As the name suggests, mucopolysaccharides. Examples are hyaluronic acid, which is also called animal cement. It occurs in animal cell coat as binding material. Then we have chondroitin present in connective tissue, heparin, which is an anticoagulant of blood. Then pectin, which is pectin protein found in the cell wall, also called as plant cement. Salts of pectin like calcium, magnesium pectates are present in middle lamella of plants. And then we have hemicellulose, which is present in the plant cell wall. Then we have peptidoglycan, which is present in bacterial cell wall, heteropolymer of NAG and NAM. And few other examples are like aloe vera and the liquidous material inside, ladyfingers, agar-agar, which are produced by the red algae used in culture media, and some isabgol boosi, if you know. Sorry, I couldn't pronounce the name correctly. It is isabgol husk boosi, which is used for the constipation patients. And gums. Gums are the natural mucopolysaccharides, like while favicol is synthetic rubber-based adhesive. Now we have a question. This question has been asked in the Bio Olympiad 2010, 12, 15, 16 and 19 also. It means this question has been asked for the 5 times in Bio Olympiad. So the question is, the molecular weight of one amino acid is 110. Then what is the molecular weight of one tetrapeptide amino acids? You know, tetrapeptide means 4 peptide bonds in it. So what will be the molecular weight of it? Now you can pause the screen and take your time. Try to solve it by your own self, then you can match the answer. Now see, the question is very straightforward. The molecular weight of one amino acid is 110 as per given in the question, and there were the molecular weight of 4 amino acid will be 440. If you multiply it by 4, you will get 440. So now how you will solve this? First you need to calculate the total weight of amino acid and subtract the weight of the water molecule released, and then you will get the answer. Try to do it. Now as we already know, when 2 amino acids react, there is 1 water molecule release out. So similarly, when there were 4 amino acids linked together, tetrapeptide amino acids linked together, there were 3 molecules of water will release out. So the molecular weight of water H2O is 18. See, we already know this. We have one
2 atoms of hydrogen, and the oxygen with the atomic weight 16, if we calculate it will be 18. So now multiply the molecular weight of one water molecule by 3, that will be 54. Then subtract the total weight of water molecule from the total weight of amino acid. 440 minus 54 will be 386. So therefore the molecular weight of tetrapeptide is 386. Answer.
Thank you everyone for being till the end. Hope you have a good day, and we'll soon come with the part 2.